Okay. Hello everyone. Thank you for joining today's Flux webinar on PhenoCam again. And I'm pleased to introduce our speakers from the Richardson Lab at Northern Arizona University, Darby Burgle, Oscar Zimmerman, and Dr. Mustafa Javadian. Oscar and Darby are PhD students, and Mustafa is a postdoc researcher in the Richardson Lab, and they are ecophysiologists who study the interactions between plants and their biotic and abiotic environments. And their research focuses on the impacts of global change on terrestrial ecosystems and feedback mechanisms between vegetation and the climate system. So today they will be demonstrating how to work with PhenoCam data, a powerful tool for tracking seasonal changes in vegetation across different ecosystems. So with that, please join me in welcoming Oscar, Darby, and Mustafa. And if you guys are ready, please take it away.
Awesome. Thank you so much for that kind introduction. So, as you guys heard, this is a part two for our PhenoCam workshop. And last week, we kind of covered what the PhenoCam network is, how you can navigate the PhenoCam website, and how you might become a part of the PhenoCam network if you're interested in that. And if this is something that you weren't able to attend last week, we have a Google Drive that is available to everybody. And I think that Mustafa can put that in the chat for you all to go check out. But within this Google Drive, you guys are going to find two PowerPoints. One of those PowerPoints is going to cover how you navigate the PhenoCam website, including our launch of the PhenoCam Explorer. So this is a little bit more interactive for you all. And then we also have our PhenoCam slides, and that kind of covers what PhenoCam is, how it got started, how you can be involved in the theory behind PhenoCams. And today we are going to be going through the post-processing of PhenoCam data, and we're going to be working in RStudio. So we'll look at some of the tools, namely PhenoCam API and PhenoCam R, and these are packages that were created to assist with the post-processing of PhenoCam data. So I'll walk through a script that's going to take about 30 to 35 minutes. And the goal here is that you guys will hopefully walk through that script with me on a cloud platform where you can launch your own RStudio. And then we will have you guys actually go through your own activity. And then at the very end, we have show and tell slides that you guys can upload some of your products from your activity to there if you're comfortable with sharing. And we'll go through that at the very end. So, Mustafa, I can't see the chat just because I'm sharing my screen, but did you
Yeah,
drop?
yeah, I just shared the link.
Okay,
Yeah.
excellent, perfect. So if everybody wants to start with selecting the link to the coding activity.document, so that is going to take you here. And please raise your hand or just unmute yourself and jump in if you have any questions on getting to this point. But once you're here, you're going to select my GitHub link. And that is going to take you to the PhenoCam workshop GitHub. And then you're going to scroll all the way down to the README, and you're going to select Launch Binder here. And this is going to open your RStudio server on the cloud. And this is going to take a couple of minutes, and I would suggest to you all that if it seems a little bit slow, that was actually really fast. This morning it was taking about 15 minutes. If it seems a little bit slow, I would select Launch Binder multiple times. So once you get to this point, and I'll give you all a minute to do this here, you guys are going to go to the lower right-hand side and you will select the Flux Workshop Environment.r data. And it's going to ask you if you want to load in that data file, and you'll say yes. And then that will give you the environment that you need to go through this coding exercise and then the activity that follows. Once you do that, you will scroll down and select the workshop activity. And this is a .rmd file. and the workshop example.rmd file. And I do want to point out that this is all in the instructions and the link to coding activity document in case I'm moving fast. And you might have to install some packages, and if it prompts you to do so, please do that. So you'll just click install here, and that will take just a moment. And then once you've made it to that point, and this will give you a bar here to show you the installation. So I'm actually going to stop here and give you guys a minute to get to this point. And it looks like somebody selected the R data, but no window showed up to click. Did you have the environment load, as in you can see all of this data that pulled in? Okay, so I would go ahead and go back over here and select this again and just click it. And then if that doesn't pop up, you may have to manually select it here and ask it to come in, but so far we've had no issues with selecting that and bringing it in. Is everybody else having success in this part?
For me, it works. Let's see if others have any comment.
Okay. Great. Okay, Kevin, I'm glad to hear that. Okay, so once we have the environment, we have our workshop activity RMD, our workshop example.rmd. From there, we want to switch it over to visual mode. This is just going to make things a lot easier to visualize, hence visual mode. Sometimes it prompts you to accept visual mode, and you can say, yes, I want to use visual mode. So we'll do this for the activity and for the example. Okay, great. And I just want to stop here for one more second to make sure everybody had success with that part. And this is important because in the next portion of this activity, those who are walking through this workshop example with me are going to be running the code chunks as I go along. So we just want to make sure that everybody is kind of in the same place as of now. If not, please go ahead and say something in the chat. But if you are where you are need to be for this portion, can you give a thumbs up in the chat too? That might be helpful.
Yeah, I see some thumbs up now.
Okay, I can't see anything, so thanks. I appreciate that. Okay, so I think we'll move on from here. And again, this is part 2 of the PhenoCam workshop. And so this is the interactive portion of the workshop. And it is going to walk us through how we connect to the PhenoCam server and use the PhenoCam API and PhenoCam R packages. And I do want to note that this code was adapted from the example code that these package creators made. But the objectives here are to examine PhenoCam metadata, to download PhenoCam time series data, to download and look at midday images from a site of interest, to extract and plot phenophases from a selected site or potentially a vegetation type that you might be interested in, and finally, to link flux data to PhenoCam data. So the first thing that we want to do here is load in all of the necessary libraries to make it through this script. And so we are going to load in our PhenoCam API package as well as our PhenoCam R package. And this is going to allow us to bring in PhenoCam site and metadata, as well as examining the regions of interest that we might be interested in. And that can also correlate to different vegetation types. So for all of these code chunks here, we are going to be using this green arrow, and all we have to do is select it. And you will see in the console here that it is going to run all of that. And I'll just stop here to make sure everything is working for folks. I see some things in the chat, but I am not monitoring that. So are we good, Mustafa, to continue on?
Yes, go ahead. Yeah,
Okay, excellent. Thanks. You can just stop me if we need to be stopped.
sure.
Okay, perfect. So the first thing that we want to do is use the PhenoCam API package to retrieve up-to-date data frame that contains all the metadata for the entire PhenoCam network. So this is going to be the get phenos function. Now, every time we use this get phenos function, it connects to our PhenoCam server. So it's always going to download the most up-to-date information, including the newest sites. So the return value is going to be a data table, which is fairly easy to work with, and it helps facilitate further data exploration. And so to do this, again, we're using our get phenos function here, and I'm assigning a data frame for this get phenos function called PhenoCurrent, because again, we are downloading all of the current information from the PhenoCam network. And then we are going to use the head function here, and we are going to call in sites to list the first 6 rows of the sites available to the PhenoCam network. And then we'll also print out the column names to get an idea of the information we get when we download this data. So we're running it here using that green arrow, and it's going to stay in this red square here until it completes running. So here again, we can see that these are the first 6 sites that are downloaded. And then we can get an idea of the metadata that is available through the PhenoCam network. So we'll have a site name, we'll have elevation, we'll have the date that it started, the date that it ended, which will be the current date if the site is still active. We'll have latitude and longitude information, whether or not the site contains infrared information, if it has flux data, mean annual precipitation, and mean annual temperature information, as well as the primary vegetation type and site description. So we might first want to focus on sites and explore their mean annual precipitation and mean annual temperature before we actually decide on an ecosystem that we want to work with, or we might just want to see, kind of the spread of the climate of the different PhenoCam sites. So in order to do this, we kind of need to have a summary table to get an idea of all of the different vegetation sites that the PhenoCam network has, so that we can use these abbreviations in our code to look at the mean annual temperature and mean annual precipitation. So this table is going to be useful throughout the example and throughout the activity, because the code for these packages requires that we use these abbreviations. So it's crucial that we have access to these abbreviations, which you can also find on the PhenoCam network under the site metadata. So the first thing that we want to do here before we plot the mean annual precipitation and mean annual temperature for all of these sites is remove any data that might be NA. So in this case, it's going to be -9999 for the PhenoCam network. So again, we're working with the PhenoCam's current data table that we just brought in. And so we're going to run this code and ask it to make a plot for all of the different vegetation types here that we selected, so we can get an idea of the climatology. So we'll run that using the green arrow again. And we can scroll down here, and let me just widen this. And so we have MAT here, MAP here, and we have our different vegetation types here. So this might help you kind of narrow down an ecosystem type that you're interested in, or potentially a vegetation type that you might be interested in. So this is really just exploratory to get started here. So once you are moving forward with PhenoCam data, you might want to extract a region of interest at a specific site. So sites can have multiple regions of interest, and usually they have these regions of interest that are given a unique identifying number. And so that can be 1,000, 2,000, 3,000. And that typically is a different region of interest of the same vegetation type. Sometimes it ends in O1 or O2, and that indicates that we had a significant shift in the camera field of view. So you can actually use the PhenoCam website to visualize where that region of interest is. So for this portion, we are going to download the phenological time series from the PhenoCam server of a site that we might be interested in. But first, we're going to download all of the sites with the regions of interest that are available.
So
And
Darby, just I'm sorry, there is just a question actually from Oscar that about that about ggplot package. So he got an error for ggplot.
Oscar, you should be able to go. Are you in the workshop example? RMD?
No, I'm actually in the workshop activity
Okay, so you should be in
file.
the workshop example RMD right
Oh,
now.
okay.
And that's going to load in your ggplot here.
Okay, thanks so much. I'll try it again.
You are welcome. So once this ggplot is loaded in the example RMD, it is already loaded and ready for the activity.
Okay, great. Thanks again.
Yep, absolutely. Any other questions at this point?
Yeah, just feel free to unmute yourself if you have any specific problem. Yeah.
Yes. Or if you need me to repeat anything, please also feel free to stop me. Okay, so we are at the step here where we want to obtain all regions of interest from the PhenoCam site. And so to do this, we are using the function called get ROIs. And again, this is a part of the PhenoCam API package. Something that I want to point out here in the code is that you will notice that this function is commented out for the time being, the actual get ROIs function. This is because in the recent months, it has not been connecting to the server and it's been timing out. So this is saved in your environment. So please do not comment this out. But it is fundamental for you guys to understand that the get ROIs function is what is bringing in all of the ROI information for the sites.
Sorry, Darby, there is just another question that I had the same error with the ggplot in the workshop example script. I'm installing it. Does it need to be? I mean, I saw that it was in the code, but
It should not need to be uninstalled. You
okay,
should have to run the libraries.
okay, cool.
Yep. So if you're having any errors like that, it indicates that this code chunk library portion here, all of them were not loaded in. So this code chunk has all of the libraries necessary for the code to work. So I would just run that. Okay, great. And again, feel free to chime in for any other questions. Okay, so here we are going to run that function, except for it has already been ran, but we're still going to explore what that function returns back to us. So we are going to use the run current chunk arrow here again. So this data frame returns the ROI name, the ROI type, whether or not the site is active. So this will give you a true false. It will give you the site name and the latitude, the longitude. And then again, it's giving us that ROI ID information now in the site name, as well as the vegetation type here. And then this indicates the actual site name itself. So what you're seeing here is that these are all underscore 1000, but again, sometimes we have underscore 2000, underscore 3000. So just note that. So now we have a data frame that has all of our ROI information. And now we can use the function called getPhenoTS, where TS indicates the time series that we're going to be downloading. And so for this, we want to focus on a specific region of interest, a specific vegetation type, and a site name. So this that we ran here is going to give us all that information. It's going to give us the ROI name, the type, the site name, and the vegetation type returned in this format so that we can put that into our line of code for our get phenos time series. So we are working with the PhenoCam ROIs data frame that we just downloaded using our get ROIs function. And for this, we are going to be working with the Kemp Natural Resources Station in Wisconsin. And this is a deciduous broadleaf forest.
There's a question, Darby. Were the ROIs drawn prior? About the ROIs?
Yes. Where are they drawn from prior?
The question is that were the ROIs drawn prior?
Yes. So the ROIs have to be drawn using the get ROIs function. So that is just how we essentially connect to our PhenoCam server and we download what ROIs are available to us and what vegetation type that ROI corresponds to. Does that make sense?
Yeah, I was wondering if the get ROIs means that you already have the ROIs, so they had to be drawn at some point.
Yes, exactly. So the get ROIs, and you'll notice that it's commented out here. So that was drawn in probably in May. And then since then, it has been timing out on the server for downloading those ROIs. So just to be safe for these workshops, I've essentially been commenting that out and saving it in the environment.
Okay, cool.
Yes. But this get ROIs, when it connects to the server, it basically will download a data frame that looks, here, I'll just show you if that's fine with you.
Yeah.
It will download a data frame that looks like this. So it'll give you the ROI name here. And again, that will correspond to the vegetation type. And then this -1000 will correspond to the ROI ID information. And then you'll get the actual site name, latitude, longitude. But the data frame that the get ROIs function returns is an important next step for filling in that information for the downloading the time series for a specific type or a specific site. Does that make sense?
Yeah.
Okay.
Thank you.
Awesome. Thanks for chiming in. Okay, perfect. So now we are downloading a time series for a site that we have identified from our get ROIs function. So
Sorry
say,
again, Darby. A question
Okay.
from
I love
Oscar.
it.
Are you saying that the ROI masks are already made? You can't necessarily draw your own ROI mask?
So you can draw your own ROI mask. If you have a PhenoCam, you can draw your own ROIs, but this is downloading pre-existing ROIs, this function. Is that helpful?
Yeah, thanks.
Okay, excellent. Thank you guys for all of your questions. I appreciate it. Okay, so moving forward here, let's say that we selected a site and that is our Kemp Natural Resources Station in Wisconsin. So that site name here is going to be this here. Our ROI ID is going to be 1000. And so with that ROI ID, our primary vegetation type is deciduous broadleaf, but it can also have a secondary vegetation type, which is our EN, which would correspond to a different ROI ID. So we're just downloading the deciduous broadleaf here. So we are going to, again, use the green arrow to run this current chunk. And this is going to download our time series information, and then we can get an idea of what that information gives us. So first, we can see that it just downloaded our site that we were interested in. It gave us our latitude, our longitude information, and our ROI type here. The active tells us whether or not this site is still actively collecting data. So this is true. And you can't see this fully all the way here, but this is our ROI name, which would be, again, the site underscore vegetation type underscore ROI type. So it would be the site name underscore DB for our abbreviation, pulling from the table above, and then underscore for our ROI type. So this is a time series that it returns for us. So we are getting GCC information here, and GCC information is typically what PhenoCam users are most interested in to track the greenness of vegetation, which is a proxy for how active our vegetation is. And then we also get our outlier information, our year and day of year information. So once we have all of that data related to our region of interest at our site of interest, which again is the Kemp Natural Resource Station in Wisconsin, we can dig a little bit further into our GCC, which is our green chromatic coordinate. And so again, this is a measure of the greenness of an area, and it serves as an indicator of the green pigment in vegetation. And so we can use that data, the GCC data, to track changes in greenness over time in a site. And so for this example, we're going to be using the GCC 90. As you can see here, GCC 90. So that corresponds to the 90th percentile of our GCC value, and that is calculated over a 3-day moving window for the ROI. You can also do a 1-day moving window, but here we're using our 3-day moving window. And so this metric is going to capture our upper range of the greenness, and it's going to minimize the influences of outliers that we might have in the data. But before we plot our GCC 90 values, we typically have to adjust the dates and convert them from factors to date format. Otherwise, your plot is not going to be very informative. So that's what we're doing here. We are taking our data that we downloaded, which again is our site and our ROI type and our vegetation type, and we are converting the date from a factor to a date format in this chunk here. And then we're just going to plot our GCC of the 90th percentile. So we are going to run the chunk here using our green arrow again. So some sites have more data than this site, but I thought that this might be a nice way to really see the GCC curves, see the rising and the falling of GCC. Sometimes you can see breaks in the GCC data here, indicating that the camera may not have been capturing data at that time. So this is just kind of a rough way to look at your GCC and the greenness activity of your vegetation at your site of interest over time. So you could isolate this to 1 year, or you could look at all year. So it really kind of depends on what you're interested in. Maybe you are interested in just a single year and what's happening at your site, or maybe you're interested in the full-time series. So this is great, right? We can see the GCC, we can see the time series at our site, but what if we want to actually peek into this forest and see what that looks like with our GCC plot? So we can actually do that, and we can access midday images and download them individually. And so these midday images typically tend to be our best images and are least affected by light conditions. And so this is why we want to download our midday images. And this is also done using the PhenoCam API package. And again, you can specify multiple years, you can specify a single month or all months, and you can specify a specific day of the month that you want to look at or a range of months that you want to look at. I see something in the chat here, so I'm just going to
No,
pause.
it was me actually, just the link.
Okay,
Sorry.
perfect. Okay, so for this, we are going to open a temporary directory. And we have to open a temporary directory through the cloud to essentially store all of the midday images that we're going to be downloading. And the function that we're using here is our download midday images function. So we're sticking with our same site here just to kind of track what our ecosystem looks like when we peek into it versus our GCC plot. So we have to give our site information. So again, that's going to be our site name here. And Y indicates the year that we're interested in. So which year you want to look at. Again, it can be a single year or it can be a series of years. And then the months that you're interested in, it can be a single month. In this case, we're looking at all months, and the day of the month that you might be interested in looking at. So this can be a single day of the month or a range. And then we're asking it to download it from the temporary directory that we opened. And then we call that directory in and ask it to download and give it this path here for our next step. So this code takes a second to run, these next two steps. And you can see this progress bar below, and it is downloading our images here. It's really helpful to have the progress bar.
I have a question. Is there a specific dating convention that is used to call these dates? Do they have to be in a specific order? Or is that just like a column in the...
Yeah, so this here, I think that the way that the midday images function works is that it's able to look at the date time information and extract what, if you give it 2022, it would be able to say, okay, from the date time, this is 2022. And then from the date time, we want all months 1 through 12 and days 15. So you can just put it in this format. You don't need to put it in a specific format for date time for this function.
Cool. Okay.
Yeah, so it makes it really accessible.
Yeah, that's great.
Awesome. So we have downloaded all of our midday images for the year 2022 for all of our months and for the month or for the day 15 for our months. Okay, so now we can demonstrate the seasonality here from the actual camera. So this is going to loop through all of our midday images and plot those images and just kind of put our GCC plot into context. So we'll run this. And this one also takes a moment here. Okay, so these are our midday images. And let me just adjust the dimensions here so you guys can see this. And so now we have each of our months listed out here across an entire year. And again, this is day 15 across all months. So you can see here that leaf out actually happened a little bit earlier than day 15 in May, and the start of our autumn colors happened a little bit earlier than day 15 of our October month. So you can kind of adjust that a little bit, but this is a nice way to physically peek inside of a ecosystem that you're interested in, to get an idea of what that camera is capturing and then how we can correlate that to the GCC values. Awesome. Okay. So now we're going to shift gears just a little bit, and we are going to talk about how we can connect flux data to PhenoCam data. And so I just want to mention here right away that the flux data that I'm going to be working with in this portion of the workshop was already downloaded using a package called Ameriflux R. And I think that there's actually a recording of this package and how to use the package on a previous webinar. So if you guys are interested in connecting PhenoCam data to flux data, the Ameriflux R package is a great resource to do that. But before we get into that, you can actually use the Flux Site Names attribute in your PhenoCam data frame that you downloaded to essentially filter every single PhenoCam site by the attribute of whether or not it contains Flux data. So this is really useful if you need to work with sites that only have corresponding Flux data. So this is going to narrow your search down if this is something that you're interested in. So we are going to ask it to spit out a data table called PhenoFlux sites. And so we're asking it to please only download the data that has Flux data, and that is our equals to true, and that it is not an NA. So we'll use our green arrow here again, and we'll run that chunk. And so again, when we use this head function here, it just spits out our first 6 rows of our data table that was downloaded, but you can always further explore the entire data table and go through all sites that are available to you. And you actually get the name of the Flux site here. And this is really helpful because this can be used in the Ameriflux R package when you are downloading that information. So you can just get the name of the Flux site here. But what if you're really only interested in a specific ecosystem or a specific vegetation type? So you can actually further filter this data frame by the primary
So
vegetation type.
Darby, there's a question. Sorry.
Yeah. Okay.
Is this still just deciduous broadleaf or all sites with flux data?
This is all sites with flux data. And next, we're further filtering by vegetation type.
Cool, thanks.
Yes, absolutely. So yeah, this chunk here is going to give you every single PhenoCam site that also has flux data. And then our next portion here is going to narrow our search a little bit further. So here we have our PhenoCam sites that contain our flux data, and then we are going to further filter it by a vegetation type that we're interested in. And I, again, am going to be focusing on the deciduous broadleaf forest. I bet at this point you guys can probably guess the type of forest that I work in. So to do this, we are going to give it the abbreviation from the table that I showed above in the beginning of this example, and that is our DB. So again, we have to say our flux data is true and our primary vegetation type is deciduous broadleaf. So here I'm asking it to spit out a different data table that is just PhenoCam sites that contain flux data that are of the deciduous broadleaf vegetation type. So again, we're going to be using our green arrow here to run that chunk. Okay, so here are the first 6 sites that contain both flux data and are of the deciduous broadleaf vegetation type. So Bartlett is actually the forest that I work in. So very convenient that it has both PhenoCam data and flux data. Okay, so now we have all of this information, but we might want to merge it together, right? And see how our flux data and our PhenoCam data can be used together. So in this case, we're going to be focusing in on the Bartlett experimental forest because this is the forest that I work in. And I already have data downloaded from this site that has been post-processed. So again, if you in the future want to merge PhenoCam data with Flux data, I would recommend using the Ameriflux R package, as well as the RReadyProc package to post-process that Ameriflux data. So again, we're going to be focusing in on the year 2018, and this data frame has already been brought in to the environment that you guys loaded in earlier. So once we merge the PhenoCam data with our flux data from the Bartlett experimental forest, we can plot our 90th percentile of our green chromatic coordinate and compare that to our gross primary productivity, and actually also plot an interactive plot to get an idea of how the GCC values correlate with our GPP values. So you guys may recognize this function that we used above, and this is our get PhenoCam time series function. So this is going to download all of the time series information that has our GCC 90th percentile for Bartlett experimental forest, and we want our 3-day here. We are converting our date from a factor to an actual date format. We are filtering our flux tower data by the year only 2018 here, and also our PhenoCam data to make sure it's year 2018. So those two merge correctly.
Sorry, Darby, there is a question in chat. For the 3-day moving window to calculate GCC 90, is there a recommended minimum number of observation over the 3 days to get reliable results?
Yeah, I think that, Oscar, do you want to speak to this? Oscar does a lot of the behind the scenes PhenoCam, so I think he might be able to answer that better.
Yeah, I mean, I think the short answer is I don't have a specific recommendation. But generally there is a trade-off between the number of images you have and sort of the smoothness of that seasonal pattern. And that's just because the more images you have, the more likely it is that the camera has captured a scene under sort of good lighting. And so I think that's that's a trade-off that you should be aware of. But the exact, minimum number of observations you need to get good data might be a site-specific thing. So that's my recommendation. I don't know if Mustafa, you have anything to add to that?
No, I think that's right, and I feel like, yeah, I mean, the more you have, the better quality probably you're gonna have. But I'm probably, yeah, I mean, probably there should be at least a few images per day, I guess, for the probably the best quality that you can get from a day, and preferably maybe midday images, I guess. But it's better to have those maybe 30-minute interval data for every days. Yeah.
Excellent. Thank you guys for chiming in. Okay, so we're at the point here where we're making sure that our PhenoCam time series data is only our 2018 data to merge correctly with our flux data. So here we are going to spit out a data frame that is our merged PhenoCam data with our flux data. And I'm just going to jump down here so you guys can get an idea of what we're going to look at after we merge that. So we are going to plot our GCC 90 and our gross primary productivity U50. So this indicates that it was the U star 50 filtration that happened in already proc. And then we're also going to look at an interactive plot of these values. So we can get an idea of how our GPP aligns with our GCC values. So we'll run this here, and this might take a moment. Okay, so I kind of want to explore this data frame again. So when we downloaded our PhenoCam time series information for Bartlett, it filtered it for the year 2018. And Marana, am I saying your name correctly?
Mirna. Thanks.
Mirna. Okay, yeah, absolutely. So I just wanted to point out that date information as well because you asked about it above. So this is how it works with that date information. So it's able to align that date time nicely. And then we can get an image count and our date of year information. And then we are merging that here with our flux tower information. So here we have our flux information, and the remainder of that information is our PhenoCam information. And so now we can look at the seasonality of our GCC as well as our GPP. So here we can see that the seasonality of the GCC as well as the GPP for the year 2018 lines up really, really nicely. So if you want to get an idea of how your camera GCC is lining up with your GPP values, you might want to plot your different GCC percentiles with your GPP. Okay, so we are going to shift gears just slightly now, and we are going to talk about phenology transition dates. So that is our start of the growing season and our end of the growing season and how we can visualize that. So we used midday images above from our site in Wisconsin to actually peek into our ecosystem and get an idea of what the growing season looks like visually, but we might want to actually quantify that. And so we can do that using our PhenoPhases function. And at this point, we're kind of switching gears into our second package, and that's called our PhenoCam R package. And so what we're going to be using here is the Burns Southern Old Black Spruce site, and that's in the Prince Albert National Park. Oscar, you might know this park. I'm not sure. But here we're going to be using our download PhenoCam function. And this is going to follow the same format as our PhenoCam API package, where we have to give the site name, the ROI ID, the frequency, which previously was 1 day or 3 day in our PhenoCam API package. And now it's just an individual number. So you could say 1 or 3. We're asking it to not do our outlier detection or our smoothing detection yet, because there's another function that will do that. And then we are assigning a data frame and asking it to read that PhenoCam file in so that it stores it nicely into a data table for us. So we will run this chunk of code here. And we can see in the console that it was downloading that information. So this is where there's actually two other functions that we can use in this package that smooths the time series and detects our outliers from the PhenoCam time series that we downloaded. And so you can do this separately to get an idea of what your data looks like before and after. And you can assign a different data frame if you want to do that comparison of before and after. Hence why I put false here in this function, just so you can separate that out and get an idea of what it actually is doing to your data. So here we're going to use that detect outliers in the smooth time series function from our PhenoCam R package. And this one also takes a moment, like the midday images. Okay, so now it's moved on to our smoothing of the time series. And once we have this data that has been made available from smoothing our time series and detecting the outliers, that's when we want to calculate and plot our phenophases, so our transition dates for spring and autumn. So this is going to be a very similar step as above, where we want to make sure that our date is in a format that is going to line up nicely with our plot. And so we can actually get day of year information to see where our phenophases are falling here. So we're going to use our phenophases function, and that is going to spit back different transition dates. So we can have transition 10, 25, or 50. In this case, I am using our transition 50, so that is our upper value of our transition dates. And we can plot that alongside our smoothed out GCC 90th percentile value to get an idea of the start and end of the season and visualize that. So we will run that chunk here. Okay, so here we can see our end and start from each year, and this kind of just gives you a quick and easy visualization of if one year may have had a shorter or longer growing season. You may have to blow up your plot a little bit more to actually see that, and you can calculate the growing season from these transition dates, but this gives you a quick and dirty idea of what the transition dates look like in your ecosystem over a given time range. So you could select a single year, or you could look at the whole time series here. So now I want to link that to the flux data and get an idea of how well our transition dates line up with our flux data, namely our gross primary productivity. And for that, we're going to come back to the Bartlett experimental forest. So we can use our phenophases function, and we can plot our GPP over time, extract our 10th, 25th, and 50th percentiles for both our spring and autumn phenophases. And that kind of gives us an idea of how GPP corresponds to periods of vegetation development and senescence. And so it kind of really tells you how well your flux data lines up with your PhenoCam data. So we're following a very similar approach here where we're, again, downloading the PhenoCam data using our download PhenoCam data function here. And again, frequency is just the number 3 for this package instead of specifying 3-day. And then we are going to filter that for the year of 2018. We're going to use the phenophases function here to extract all of our percentiles of our transition dates. And then we are going to look at our transition dates here. So our rising is our spring transition dates. Our falling is our autumn transition dates. And then we are going to assign it as a date. And then from there, because it's in Unix format, so we have to assign a date. So you can actually have a day of year information and get an idea of where that falls on your plot. And then we are going to plot those transition dates on top of our GPP data. So let's go ahead and run this chunk and get an idea of where those transition dates land. And this one is a bit more code. so it might take a moment. And this is our last step in the example here, and then we can move on to the activity. But I do want to make sure that everybody has the libraries correctly loaded for the activity so that it goes smoothly.
So while you're running this part, I just wanted to mention that the webinar recording will be available on the AmeriFlux YouTube channel. I just put the link in the chat, so just don't worry if you didn't follow any part or something.
Yes, and also my GitHub is publicly available, and you guys can launch this binder anytime and look at the scripts anytime. You can also fork my repository and bring it into your own RStudio and look at it as well. And I think the instructions are pretty detailed for you guys to be able to follow. Okay, great. So now we are looking at our GPP at the Bartlett Experimental Forest for the year 2018, and we're looking at all of our different phenophases. So our 25th, our 10th, our 25th, and our 50th percentiles for our start and our end. And so this can kind of give you an idea of what you might want to use for your ecosystem. So, and Oscar might be able to speak to this a little bit too, but for different ecosystems, there are different transition dates that may more closely align with the actual phenology in your ecosystem with the GPP flux data. So this can kind of give you a visualization of what you might want to use and which one of these transition dates correlates most closely with your GPP values for the start and end of the growing season. Awesome. So that is going to conclude the example. And if everybody could quickly scroll to the top of the example, that would be fantastic, because I want to make sure that before we move on, everybody has run this chunk of code here. And that is going to pull in all of the libraries necessary for you guys to use. Otherwise, you might get an error that a function is not available. And that just means that a package wasn't loaded in. So
It's a
let's
quick
run
question.
that. Yeah,
Can you hear me good? Because I have microphone issues.
I can hear you just fine.
All right, perfect. I'm running the code right now. I was trying to watch you do your lecture and just focus on that, but I started running the code, and I'm having troubles getting ROIs. I keep having issues right there, and I know you mentioned something earlier, but I forgot.
Great. So it sounds like what might not be in your RStudio yet is the environment.
That's
And
what you're saying. Okay.
yes, yeah. So if you could just go to the lower right-hand side of your RStudio and select this Flux Workshop environment.r data, and it will say, do you want to bring in this data? And you will say yes. It should work after that because that data is already available in the environment.
Perfect. Doing it right now. Thank you.
Yep, absolutely. Okay, so once everybody has run this chunk of code here to make sure all of your libraries are available, so your functions work, you guys will be working on the workshop activity. And I just want to talk about this for one moment to alleviate some common points of confusion that we typically have here. But you guys are going to essentially read through these different portions of the code chunks. And if you see run, all you need to do is run the code chunk. Other times I will ask you to insert some information. So in this case, I'm asking you to choose two vegetation types from the table. So for example, it could be GR, it could be EN, and where you see vegetation type 1 and vegetation type 2, you guys are going to actually replace that information in the code here. But if there are quotation marks in any of these code chunks here, make sure you leave those. Otherwise, your R code is not going to understand that you are giving it a vegetation abbreviation. So that's usually what we run into, is that things have not been replaced where they were asked to be replaced, or the quotations were removed. This one can be confusing sometimes where it says ROI name, site, ROI type. You guys are going to replace that information and give it the actual ROI name, the actual site, and the actual ROI type that you'll get from the My ROIs information. So I think that's really all the information you guys need to alleviate some of the most common hiccups that we run into. And we have some folks that have joined us today that are going to help troubleshoot any issues that we might have with this coding activity. So we'll probably send folks to breakout rooms if we have a lot of questions. Otherwise, we might just stay in the main room. And then the drive that Mustafa has been sharing quite consistently also has a show and tell a slide deck. If you guys want to share anything that you added, sorry, the Zoom bar is getting in the way right now, but that's going to be our workshop show and tell slides. And we have some examples that folks from previous workshops have uploaded. So if we have time at the end, we can kind of go through some of the products that you guys were able to produce. Awesome. And then as you guys work through the activity, just go ahead and chime in and let us know if you need anything at all.
So there's a question in the chat, how to draw your own ROIs, any instructions to follow. So basically, the ROIs are selected by the PIs of the sites, and basically these packages are just getting the ROI information from each site. And but there are some other packages like PhenoPix and other packages that you can draw your own ROIs and and working on kind of repeated RGB images and draw your own ROIs. But if you want to use PhenoCam ROIs, it basically those ROIs that are available for those on the PhenoCam website for each site. And basically, sometimes a PI of a site may ask us to change the ROI or update the ROI, and then we just change the ROI accordingly.
Thanks for answering that.
I have a sort of related question. So this package is for querying from the PhenoCam network pretty much exclusively. So if you have a different kind of uncompatible camera and sort of locally saved images, I'm working with images on my computer, does the PhenoCam API service that at all, or not really?
I think it depends on if you're a part of the PhenoCam network or not. So you can be a type 1, type 2, or type 3 site, and that kind of breaks down whether or not you follow the best practices that PhenoCam suggests that you use. But say you have your own camera that's not a part of the PhenoCam network, my suggestion to you there would be to go to the PhenoCam API or PhenoCam R GitHub and just fork that package and bring it into your own RStudio, and you can just save those functions into your local RStudio, give them a different name if you want to, and then adjust the names in the functions to the names in your data frame. And then you could essentially keep all of the source code the same, but apply it to your camera.
Okay, cool.
Oscar might also have another response too. He just unmuted.
Yeah, nice. Good noticing that, Darby. Yeah, Mustafa just made a comment in the chat with other resources that might be useful for those kinds of things.
Yeah, I just shared the PhenoPix package, the paper that is talking about the PhenoPix package that basically gives you the opportunity to, even if you take images with your phone and you have RGB images, you can use this package, and it's kind of independent from PhenoCam network.
Cool. Yeah, I'm using PhenoPix right now.
Yeah, that is also another, yeah.
And so are you suggesting kind of using both PhenoPix and then to draw the ROI, say, and then um using the PhenoCam API or PhenoCam R?
So I probably think that if you don't want to like upload your data to PhenoCam network, you can use this PhenoPix package and you can just go with that, because this package is kind of powerful package, and there are so many papers that have used this package for RGB images with different kind of platforms, like from Raspberry Pis, from different type of even like phone images, different things. So probably this package should be enough, but there is also another way that maybe you would be able to like have a site in like PhenoCam, and then we can process those images for you on the PhenoCam website, and you can just download the data with these packages that Darby was talking about that here.
Gotcha. Thank you very much.
Thank you.
So Darby, you said that you have a helper here to help troubleshoot some of the R coding.
Yes, well, you are one of those, and Perry.
Yeah, I'm here if needed.
So Perry Jambuzzi, who's on the Zoom call, is a grad student in our lab working with PhenoCam data and doing modeling of autumn phenology. And so Perry's here too to help us out today.
Yeah, happy to help if possible.
And we're happy to have you.
And if any folks are feeling like the activity is a big chunk to work through, you guys can always just focus on what interests you the most. For example, plotting a GCC curve from a site you're interested in, or downloading midday images. You certainly do not need to work through this entire script. And again, I just want to mention that this is available to all of you guys to look back on if you need to run through any of this again. Mustafa, do you think that we should plan on looping back around at 11:15 and going through the slideshow, or should we give a little more time?
Maybe we can give them some time to like maybe run the exercise, and then
Okay.
yeah,
So maybe 11:20.
maybe, yeah, that would be great.
Okay, sounds good. So we will just be on standby for you guys, if anybody needs help, and then we will loop back around at 11:20 to see if anybody has uploaded things into the slide deck for the show and tell, and we will discuss at that point.
Yeah, and just feel free to unmute yourself if you have any questions.
I have a question.
Great, yeah.
So with this RStudio server, are we able to knit? Like I want to knit to Microsoft Word, just so I can have just a
Mm-hmm.
yeah.
Yeah. So great question. And we've had this question before, and I have to admit that I have not tried to knit it or push anything to my own computer, but I can bring things in from my own computer. So I think that you would be able to push something to your computer.
Yeah, perfect. Thank you.
Yeah.
Yeah, I got a question too for this first one. I'm not able to pull up a ggplot. Maybe I should hop in a help
Yeah.
room on my screen.
Do you want to just move over to the workshop example really quickly, the R markdown, instead of the activity? And then in that first code chunk, if you just want to run this really quickly, I think it's just a package loading issue.
Wait. Oh, yeah. i.e. the ggplot didn't load.
Yeah, so if that is not loading, then you probably just have to type install.packages and then parentheses in quotes ggplot2.
It should be installed. Were you able to run the ggplot now?
No.
Okay.
Okay, hang on.
I am going to
Hang on.
I'm going to paste this into the chat. And then if you just want to run this line of code in the chunk, that should alleviate the error.
I think Xiangmin has a question too.
Great.
Yeah, so I'm trying to just launch Binder and then I cannot add that Flux workshop environment data. So I just clicked it, but there's no window popped out for me to click yes. So I tried Chrome and I tried IE browser, but they all do not show up that window. So I could not add the file.
That is
I'm
so interesting. Do you want to share your screen? Can we
sure,
have
sure,
access? Okay,
sure.
here. Let me stop sharing mine really
A
quickly.
similar issue.
Similar issue?
I had a similar issue where I just check marked it and thought it would run, but I just double clicked it and it populated.
Oh, you just double clicked. Okay. So
Okay. Yeah, yeah. So okay, now there's something wrong.
great. Looks like
But
it's
when
launched.
I go to click it and I click here,
Mhm.
okay, now it seems this window didn't show early.
Okay, so yeah, this will take a moment to launch here. And then as soon as that launches, I'm going to share my screen again.
Okay.
As soon as that launches, it's going to pull up with something that looks like my Zoom bar keeps getting in the way. Something that looks
No
like
hurry, no
this.
hurry.
Uh-huh. And then once you're here, that's when you're going to want, it sounds like some people only had to click it once. You may need to double click this
Okay,
for it
okay.
to work. And then let us know if that works.
Sure. Thank you.
Yeah, thank you.
I think Brandon had a question too. If you, I don't know.
Yes, Brandon raised his hand, but I think, was it about the environment?
Oh, okay.
Okay. Brandon, are you all good?
I think I'm having audio issues again, but yeah, it was about the environment.
Okay, awesome. Thank you.
And so now the binder seems does not working.
It's not launching the RStudio.
Right.
Okay, so go ahead and and close the ones that are not launching, and then try to launch it multiple times. The more times you launch it, typically the faster it opens your RStudio server.
Okay. Okay, sure. Thank you so
It's
much.
Yeah,
Mhm.
absolutely. I mean, this is one of the best cloud platforms we found. However, it can be really inconsistent in the amount of time it takes to launch a server. Sometimes it's 10 minutes, sometimes it takes less than 5 seconds.
Yeah. Okay. Mhm. So now I feel it has something wrong. So probably I can share my
Let
screen.
me stop sharing my screen
Yeah.
here. And then if you want to
Okay.
share your screen,
Yeah.
I can look at that. Okay, yeah, so it's still launching. I don't see.
So there's some error happened here. I'm not sure whether it's normal or not. Okay.
Oh, okay. Oh, let's see. Why don't you? So let's exit out of this binder again, and then go to the link to the coding activity and select that link a couple of times.
This one?
Yep. And then I would open it. I would launch this binder, and then let's launch a few more binders here and see if we can get something to launch quickly.
Okay, if this does not work,
And maybe if you can try on your incognito mode, because that sometimes help,
Oh, sure. Good idea.
That is a good idea.
Okay, this time it seems to work. There's no error show up.
Great. And let's see. Oh, it looks like it wants to launch. Yep, there it goes. Yay, good.
Oh, nice.
Okay, awesome.
Nice. Thank you so
So
much.
now
So-
you'll want to, yep, so actually select it, unselect that and then click on that. Yep, right there. And then say
Oh,
yes.
okay, sure.
Yeah,
Now,
there you go. Perfect. And then
now
you'll
it's
want to bring in the workshop activity and you'll want to click it again. Yep. Perfect. And then
okay.
the workshop example. Yep. And then go ahead and scroll. Yeah, you'll want to install those. And then go ahead and scroll down a little bit on the workshop example. And then, up a little bit, sorry. Up one more little chunk. So you'll want to run the code chunk that has all the libraries.
Yeah, right?
Yep, exactly.
Okay, sure.
And then you can move on to the workshop activity after that. Oh, it's still installing your ggplot. That's why I didn't load it.
Okay,
So
I
as soon as it's done
can
installing.
go here to install it.
It does look like it's installing it in the background right now.
Oh, nice.
Yep, so
Okay.
I think you'll be fine. Just as soon as that progress bar completes, you should be able to then load ggplot library.
Okay, great. Thank you
Awesome.
so much.
Yeah, absolutely. Yeah, so it looks like that finished and you can run the library chunk again. Perfect. Great. And now you can kind of explore the workshop activity or the example if you want to, either or.
Sure. Okay, great. Let me just stop share.
Awesome.
Yeah, nice.
And it would be great if you have any results or whatever that you find interesting to just share it in our show and tell slides in the Google Drive folder that we shared. Whatever you find interesting, that would be cool.
yeah, we will loop around in about 5 minutes to hopefully look at some of that if anybody was able to upload some show and tell information from the activity. I do see the question here in the chat. Something that I have found was helpful for some folks in the past to identify their site name. Sometimes it's an incorrect site name. Is going to the PhenoCam website, and then going to the gallery, and then searching a site there if you are having a hard time getting the correct site name. Otherwise, you can search through the data frame that you downloaded and look through all of the site names from PhenoCam. But this is a little bit more streamlined and is great because you can also use the PhenoCam website while you're
Yeah.
on code.
Yeah, and even on the top of that page, there's a map button that you can even go through the map and then see what is the closest maybe PhenoCam to your area or whatever.
For example, say we went here, and then this will give us our site name. Wow. And this would be our site name here. We could also, while we're on this, we could also go to the site metadata here, and that gives us our vegetation abbreviation for the primary vegetation type, as well as our ROI ID. So this is -1000 here. So it looks like we have some things that have been uploaded to the show and tell, but I will give you guys just a few more minutes if anybody is interested in sharing. Okay, folks, so we have about 8 minutes left here for this workshop. And it looks like there are still some things being added to the slide, but if you guys would be comfortable just starting with the first one here, if anybody wants to sort of speak to a site that they chose or chime in, that would be excellent. And you can just kind of unmute yourself and share what you were able to do here.
So I can speak for my example that I use Morgan Monroe because you're showing it right now. But yeah, this is a site that I've been working mostly for my PhD study, which is located in Indiana. And this is a deciduous broadleaf forest, kind of really typical site at with the yeah, with major species of tulip poplar, sugar maple, and oak species, and yeah, I think it is showing pretty typical pattern of greenness over time. Yeah, seems to be doing really great job.
So Kung, when you were doing work at this site, how many years of data did it have?
So back then, so Tower started collecting data since 1998, I believe. But right now it is showing the data from like 2009 or so. Yeah, but yeah, I used to look at like, the data from early 2000 to early 2010, but yeah, for my study.
So the PhenoCam data and the Ameriflux data has come a long way since.
Yeah, seems like so. Yeah, I don't really know, yeah, since when they have installed PhenoCam and yeah, started collecting data, but pretty impressive having really long record.
Thanks for sharing, Kung.
Yeah,
Thank you.
for sharing. Okay, folks, I am going to go to the next slide here. And if somebody wants to speak to this site, that would be awesome.
Hi, yeah. So I just selected that type. It's in Brazil because the vegetation there is like I think it resembles what we can find in my country in Ecuador, like evergreen broadleaf forest. So yeah, it was just interesting to see that the pattern, the GCC signal there. So I don't know if that's like maybe some issues with the images or yeah. I mean, yeah, it was just interesting to see.
Yeah,
Thanks, Fran.
oops, sorry, Oscar.
Oh, sorry. I was just going to say thanks, Francisco, for sharing. And you're right, it is sort of an atypical pattern in terms of maybe the long-term pattern's a little atypical. It's almost as if they cut down the plantation trees at one point and the plantation grew back. I'm not sure.
Oh yeah, that makes sense. Good point.
What year did you select for the midday images?
Last year, so 2023.
Great, yeah, thank you. Okay, we will go to the next slide here. All right.
Hi. Hello, this is me. Hello, guys. Thank you for the workshop. Yeah, I was there the beginning of this year in a workshop at your university, and now I'm glad to have it again because I had the opportunity to see this again. So now I am showing data from my site at La Selva in Costa Rica. So it's an evergreen broadleaf forest. It rains a lot throughout the year. We only have some months of the year. I'm not showing the entire year here because it's not available, but as you can see, there is always greenness. But it's interesting to note in the time series plot that there are some this sharp decreases in GCC, and I wonder if maybe that day was like huge rain or something that could not, the camera could not capture the greenness for some reason, you know, like July, for example, usually is quite humid. But yeah, interesting.
Thank you so much for sharing. And that's where I think, you know, having the availability to actually go to the PhenoCam website and looking at the images from when you see this drop in GCC can be really informative to kind of put that data into context and get an idea of what might be happening there. Okay. We have another plot here of GCC.
Okay, yeah, this site is in Lincoln, Nebraska. So it's a prairie site, and I just analyzed it. I just copied your data and to just repeat the analysis on this site. So it shows clear pattern of growing season from 2015 to 2017. But I think this site is always optional, but I'm not sure why it only shows the data from 2015 to 2017. I I I expect it will show data until now. Maybe I need to revise my code a little bit.
It could also be because the data is split over more than one ROI.
Oh, probably, yeah.
That is exactly what I was going to say. So the PhenoCam website could help you answer that question. If you have the site name, you can go to the PhenoCam network and it will actually show you under the metadata and on the right-hand side how many ROIs are available for this site. And you can explore those different ROIs.
Okay, so now I can share you this this link for the site, and I'm not sure how to check the ROI.
Oh, yeah.
Mm,
Do you want to share your screen again for that?
sure.
Okay, I'll stop sharing here.
So I'm going to. Okay, so this site is our site. So here there are four ROIs.
Exactly.
Okay. So now let me see. Oh, okay.
Yep, so that's only showing the 2 years there, it looks like.
So now if I want to use all those ROIs, I need to go back to my code to somewhere here to change it here, right?
Yeah,
No?
so you would have to go up one and make sure that the data that you're downloading is the ROI 2000 for ROI.
Here, right?
Yep, right there.
Okay.
And then that will, I mean, you're going to replace the 1000 data frame that you downloaded. So you could just, you know, run that code again and assign a different data frame name to it.
Okay, so now DB 1000 and GR 1000, let me see. But here, this DB, okay, now I understand. So
Yeah.
what's the DB means and what's the GR means?
So those are the primary vegetation types. So it means that the region of interest is capturing 2 different vegetation types.
Oh, okay, nice. Thank you so much.
Yeah, absolutely. Thank you. Okay, folks, I think, is that everything we had in the show and tell? I think it was.
I think so, yeah.
Okay, perfect. Well, that concludes PhenoCam Workshop Part 2, and I just want to mention one more time that all of the code in my GitHub and the slides from part 1 are all available to you guys to look back on. And a recording of this workshop as well as the first workshop is going to be available on YouTube for everybody. And you guys can always email us in the PhenoCam Network team if you have any questions. And I think our emails are available on the slide deck, and we can also paste those into the chat before we head out for the day.
Thank you, Darby, and
Thank you
Mustafa
so
and
much.
Oscar, everyone from Richardson Lab. I mean, I really appreciate the really valuable resources from this lab they developed and shared with us today. And I believe this is really going to be a really great asset to even the science community broadly. So yeah, again, this session is recorded and will be uploaded to the YouTube channel, Ameriflux YouTube channel in the coming days. And anything else to share, like Darby?
I just wanted
Mustafa?
to thank you and the Ameriflux management team for helping us to organize this seminar. So it's very cool, and thank you so much for everyone. Thank you so much everyone that joined us today and last week. And just let us know if you have any more questions by email or whatever. And also, yeah, as Kung said, the video is gonna be online available and also on the PhenoCam website too.
Awesome. Thank you so much. And with that, thank you, everyone, and have a wonderful weekend. Bye.
Thanks, everybody.
Thank you. Bye bye.